So this is chapter 19, in which we're going to talk about blood. So we're going to start with an introduction and talk about the overall cardiovascular system and introduce you to some fluid terminology. So the next 3 chapters will be covering the cardiovascular system. And if you remember from chapter 1, when we looked at all of the organ systems and their functions, we went over the functions of the cardiovascular system, which includes distributing blood cells, water, and dissolved materials, including nutrients, waste products, oxygen, and carbon dioxide. It also distributes heat and assists in control of body temperature. So the cardiovascular system is made up of blood, the heart, and the blood vessels. So in this chapter, chapter 19, we're going to talk about the blood. And if you remember from chapter 4 last semester, blood is a type of fluid connective tissue. And so this is the fluid that circulates within the cardiovascular system. In chapter 20, we're going to look at the heart, which is the pump that is responsible for circulating the fluid within the cardiovascular system. And in chapter 21, we're going to look at the blood vessels, which are the pipes through which the fluid flows. And just a heads up, chapter 20 and chapter 21 are going to have more physiology in them than your average chapter. Before we get started with chapter 19, let me go over some terms that describe various fluids within the body. We're going to start seeing these terms a lot as we move forward, and I know sometimes students get these mixed up, so I wanted to give you this slide at the beginning of the lecture so you could come back to it easily in the future if you need to refresh your memory. So intracellular fluid is the fluid found inside the cells. So we talked about this last semester as being the liquid component of the cytoplasm, which was known as the cytosol. Then there is extracellular fluid, which is any fluid that is outside of the cells. But since there are a lot of different compartments in the body outside the cells, we can further divide extracellular fluid into subcategories. So extracellular fluid includes the plasma, which is the fluid component of blood. So this will be only found inside of blood vessels. You could also say it is intravascular fluid, but I don't usually see this term very often, so I just wrote blood here because plasma is the fluid component of the blood. Then you also have interstitial fluid, which is the fluid in the tissues. So think of this fluid as being outside of the cells, but also outside of the blood vessels. So one of the focuses coming up in this cardiovascular system section is how solutes and other items move between the blood plasma and the interstitial fluid. So now you can better visualize these two separate compartments of fluid. In this section, we're going to look at the functions of blood, the characteristics of blood, and the components of blood. So we'll start with the functions. Blood functions to transport dissolved gases, nutrients, hormones, and waste throughout the body. So as far as the gases go, the 2 primary gases are going to be oxygen, which is carried from the lungs to the cells, and carbon dioxide, which is carried from the cells to the lungs for excretion. So why do we have to care about this movement of oxygen and carbon dioxide anyway? Well, I wanted to refresh your memory from something else you learned last semester in chapter 3 when we talked about the cells. All cells have an organelle called mitochondria, which is the energy factory for the cell. So the mitochondria makes ATP, which is the energy currency. And ATP is used to do all of the jobs. Like last semester, you learned the importance of ATP in muscle contraction and in like the sodium-potassium pump, which maintains sodium and potassium ion concentrations. But in order for the mitochondria to make ATP, it requires oxygen and glucose. And it takes the oxygen and glucose, uses it to make the ATP, and then 2 of the side products or waste products are carbon dioxide and water. And so the sole reason that we need oxygen is for the mitochondria and all of our trillions of cells to be able to make ATP. And the sole reason that we have to worry about getting rid of CO2 as a waste is because all of our cells are producing CO2 as a byproduct of the ATP making process. So this gets more important the further we go along the semester. For example, in the respiratory system, the entire reason we have a respiratory system is just to get the oxygen and get rid of the CO2. So it all boils down to this right here. And then even later, we're going to talk about the acid-base balance in the body, and CO2 or carbon dioxide plays a large role in that as well. So remember, it all goes back to the individual cells of the body needing oxygen to make ATP and producing carbon dioxide as a byproduct in the process. So in addition to transporting the gases, blood will also transport nutrients from the digestive system, so the stuff that we eat and drink, and also it can take nutrients out of storage and circulate them around, like from adipose tissue or our glycogen stores. Blood can also transport hormones from the endocrine glands, as we discussed in the last chapter, and then all sorts of waste products, including carbon dioxide, which is considered a waste product as well. Another major function of blood is that it regulates the pH and ion concentrations in the interstitial fluids. And as we discussed in the intro section, the interstitial fluid is the fluid in the tissues, which is outside of the blood vessels, but also outside of the cells. Blood can also function to restrict fluid loss at injury sites. So our blood is able to form clots through the actions of platelets, which act as temporary patches to prevent excessive fluid loss from the body. And we're also going to cover that later in this chapter. Blood also helps us defend against toxins and pathogens. So blood contains both white blood cells and antibodies. We'll introduce you to the different types of white blood cells in this chapter, but we're going to go into much more detail in how they do their jobs and what antibodies are when we get to chapter 22. And the final function for blood is that it helps to stabilize body temperature. So when you get cold, for example, the blood vessels in your skin and your extremities, like your hands and your feet, will constrict, which reduces blood flow to those areas. That's why you get cold hands and cold feet, and to keep the warmth inside where your major organs are. And then conversely, when you get too hot, the blood vessels in your hands and skin and feet can dilate, which allows you to expend extra energy into the environment. So now let's look at the physical characteristics of whole blood. The temperature of your blood is actually slightly higher than your normal body temperature. So blood has an average temperature of 38 degrees Celsius, whereas body temperature has an average of 37 degrees Celsius. Blood is also 5 times as viscous as water, and that means that it is 5 times as sticky, 5 times as cohesive, and 5 times resistant to flow as water is. And so basically summed up, you can say that the old adage is actually true. Blood really is thicker than water. So the extra viscosity that blood has is actually due to the cells and the protein content in the blood, which is what makes it thicker than water. Blood has a pH between 7.35 and 7.45, so it is slightly alkaline. So a neutral pH is a pH of 7, so anything above 7 would be in the alkaline range. Also notice that this is a very narrow range, and so we're going to talk about later in the semester how the body can keep this range within this narrow parameter. And then blood volume is usually 7% of your body weight in kilograms. So a 75 kilogram person, which would be about 165 pounds, would have about 5.25 liters or 5.4 quarts of blood in their body. If we look at whole blood, it is composed of 2 major components. The first one is the liquid part of the blood, which is called the plasma. So this makes up an average of about 55% of your blood. And notice that plasma is mostly water. So plasma is made up of 92% water. But the plasma proteins, which are 7% of plasma, are also important, and we're going to cover those in a moment. The other solutes are those dissolved solutes we mentioned earlier, like the gases, the nutrients, the hormones, the waste products, etc. The other part of whole blood is what we call the formed elements. And the formed elements are all the cells and cell parts that are within blood. This is going to be mostly red blood cells. So notice that red blood cells make up 99.9% of your formed elements. But you also have some white blood cells and some platelets, which are pieces of cells as well. Formed elements are going to be a focus of most of the rest of the chapter, so I'm not going to say much more about them now because we're going to talk more about them in upcoming sections. A couple of clinically relevant terms before we move on. The hematocrit is a percentage of formed elements in a sample of blood. A lot of people will also use the hematocrit as a stand-in for the number of red blood cells that you have because, remember, of the formed elements, red blood cells make up 99.9%. So if you're measuring the percentage of formed elements, most of those are going to be red blood cells. But the official definition of hematocrit is the percentage of formed elements, which is all cells and cell parts in a sample of blood. This is also called the packed cell volume, or PCV. In adult men, the hematocrit is normally 46, so this would mean 46% formed elements, which would mean 54% plasma. In adult women, the hematocrit is normally 42, so again, this would be 42% formed elements, which would be 58% plasma. The reason that men normally have a higher hematocrit than women is because men have a much larger androgen circulation, and testosterone is the primary androgen, and androgens do stimulate red blood cell production. Another term is a plasma expander. This is a solution to increase blood volume temporarily after a blood loss. So we'll talk a little bit later about the dangers of hemorrhage and blood loss. One of those dangers is a sudden drop in blood pressure. And so a plasma expander can buy time while waiting on a blood transfusion. So while they're blood typing the blood to see what type it is and cross-matching it to make sure they get the correct blood to transfuse, they can add a plasma expander by IV in the meantime to actually restore blood pressure back to normal until the blood transfusion can be done. And we're going to talk about blood typing a little bit later this chapter as well. Plasma expanders usually contain large carbohydrates to maintain the osmotic balance of blood. It does not help with oxygen carrying ability because it doesn't contain any red blood cells. So as I mentioned, this is just a stopgap to buy time until the patient can actually get a blood transfusion. In this section, we're going to look at plasma and the plasma proteins. So again, remember that plasma makes up about 55% of whole blood. And so plasma is the non-formed element portion of the blood. So if you take out the red blood cells, the white blood cells, and the platelets, you're left with the plasma. Plasma is 92% water and 1% dissolved solutes, which, as I mentioned before, is going to be your gases, your nutrients, your waste products, your hormones, etc. And then plasma is 7% plasma proteins. So we're going to talk in more detail about what these plasma proteins are. So about 100 milliliters of plasma will contain about 7.6 grams of protein. So that's why we say it's about 7% plasma proteins. There is a much higher protein content in the blood than in the interstitial fluid. And that is because the large size of the proteins means they can't cross the capillary walls to get into the interstitial fluid. So this is one way that the plasma of the blood differs from the interstitial fluid, is in the amount of protein you can find in it. More than 90% of the plasma proteins are made by the liver. So if you have liver disease, this can adversely affect your blood composition and can even do things like interfere with your clotting if the liver is unable to make the clotting proteins. So as we're going to see as we move through the semester, the liver does a lot of things. Overall, the liver has about 200 different functions. So now let's look at the three major types of plasma proteins, with the first group being albumin. Albumin makes up the majority, about 60% of your plasma proteins. These are your major contributors to osmotic pressure, so making sure that the blood has the right concentration of solutes for osmosis to occur. And the function of the albumin is to transport fatty acids and hormones. So back in chapter 18, we talked about how hormones could be carried by proteins. And when the hormones are carried by proteins, they're called bound hormones. So in chapter 18, we talked about free versus bound hormones. Now we're actually looking at the proteins that do the transporting. So an albumin molecule would be an example of a transport protein. So it can bind to hormones, it can bind to other things like fatty acids as well. The globulins are the next group of plasma proteins. This is the second most common. About 35% of your plasma proteins are globulins. Antibodies, another name for your antibodies, is actually immunoglobulins. So antibodies are a type of globulin. We're mentioning that here, and we won't really talk any more about antibodies until we get to chapter 22. There are also transport globulins, and they work very similar to the way the transport albumins work. So they can bind to hormones. They can also bind to ions, both things like metal ions and other types of ions. One example of a transport globulin you've probably heard of, even though you're not aware of it yet, is an apolipoprotein or a lipoprotein. These transport globulins help to transport lipids, which are fats, through the blood. And if you remember again from last semester, lipids are hydrophobic, meaning they don't mix well with water. Well, plasma is 92% water, so these lipids wouldn't travel well in the blood unless they are carried by a protein. So here's a picture of a lipoprotein. So a lipoprotein has this big protein molecule that wraps around it called an apolipoprotein. And then you have a single layer of phospholipids. It's not a bilayer like our membranes, but this is rather a single layer. And then on the inside of these, you have these collections of cholesterols and triglycerides, which are both types of fatty molecules. Now, I said you've probably heard of these. These are LDL and HDL. So a low-density lipoprotein is LDL, and a high-density lipoprotein is HDL. You've probably had discussions with your doctors about these. So sometimes they call LDL the good cholesterol. LDL is the bad cholesterol, and HDL is the good cholesterol. And the low-density lipoprotein means that this one is more fat and less protein, and the HDL is more protein and less fat, which is why the HDL is the better of the two kinds, because it's actually transporting less fat through the blood. The third type of plasma protein is fibrinogen. This makes up 5% of your plasma proteins, and fibrinogen functions in the blood clotting process, which we're going to look at detail later in the chapter, but just for now I'm going to go ahead and introduce it to you. So as part of the clotting process, fibrinogen, which is soluble, meaning it dissolves in blood and you're not really even aware that it's there because it's dissolved. During the clotting process, fibrinogen can be converted by an enzyme into fibrin, which is an insoluble fiber which actually forms the clot itself. And again, we'll look at a little bit more detail later. So some more clinical terminology to know. Plasma, as we've already mentioned, is the blood with the formed elements removed. So if you take a vial of blood and you spin it down in a centrifuge, all of the formed elements are going to go to the bottom of the test tube, and all of the clear part at the top will be the plasma. However, something else that is commonly used in the clinical setting is serum. Serum is when you take the plasma and you remove the fibrinogen. So you remove its ability to clot, and then you call it serum. So this would be plasma with the fibrinogen removed. So here's a picture to help you see the difference. So if I want plasma, then I'm going to have all of the formed elements separate down here, the white blood cells and platelets, the red blood cells, and then plasma will be left at the top. In order to collect this, you have to have a test tube that has an anticoagulant in it to prevent blood clotting from happening. And then if you do want serum, then you let the blood clot. It all goes down to the bottom of the test tube in a giant clot. And because the fibrinogen was used in the clotting process, now you have serum, which is fibrinogen free. So just a little piece of knowledge for you, and I'm not going to go into detail here, but the reason why they take your blood into multiple different containers at the doctor's office or hospital, and each container has a different colored lid, is because some of these are coated with anticoagulants so that they can get plasma. Some of them are not, so that they can get serum, and some of them are coated with other substances that help them with other specific types of blood tests. So depending on what the tube is coated in is what the color codes mean. And if you're going on to nursing school, you'll learn all about that along that pathway. So in this section, we're going to look at red blood cells, which are also called erythrocytes. So if you go back to your root words, erythro means red and cyte means cell. So this is the official name for a red blood cell is an erythrocyte. So we're going to talk about the structure-function relationship, and we're going to talk about hemoglobin in this section. So the red blood cell or erythrocyte is the most abundant formed element. So as we mentioned before, it makes up 99.9% of all of the formed elements. It is one of the most specialized human cells. So it's going to look very different than that typical human cell that we looked at last semester in chapter 3. In fact, the red blood cell has no nucleus and it has no organelles. It's mainly just a protein called hemoglobin and a bit of cytoskeleton. So its structure is what we call a biconcave disc. So it has this disc shape, and it's got an impression on both sides, so that's why it's called biconcave, which gives it like this valley region in the middle. Without a nucleus and without ribosomes, red blood cells cannot make new proteins. So last semester we talked about the protein making pathway and how it started with genes in the nucleus, and then the RNA carried the message out to the ribosomes. Well, red blood cells can't do any of that. So as a result, when they get worn out and break down, they can't make repairs to themselves. So the lifespan of a red blood cell is only about 120 days or around 4 months. And we're going to talk in another section about how you're constantly making new red blood cells. The red blood cell is a hard worker though. So in those 120 days, a single red blood cell will travel about 700 miles around inside your body. Also note that they have no organelles, which means they also have no mitochondria. And as we discussed on an earlier slide, mitochondria use oxygen to make ATP. And so it's important for red blood cells not to have mitochondria, because otherwise the mitochondria would use up all the oxygen that the red blood cell is supposed to be carrying out to our other cells. So the fact that the red blood cells have no mitochondria means that it's able to take that oxygen and carry it out to the other cells of our body. So let's look at what we call the structure-function relationship of the red blood cell. So the biconcave disc structure and flexible plasma membrane have 3 important effects on the function of a red blood cell. First, it gives red blood cells a large surface area to volume ratio. And what that means is that with the biconcave shape, as shown on the right side, you've got a lot more surface, or surface area compared to the volume on the inside of the cell. And this is what it might look like if we had a completely round cell that was like a sphere. And all of these little dark molecules in here are just molecules of hemoglobin, because that's pretty much all red blood cells are packets of hemoglobin. And remember that hemoglobin is carrying oxygen. We'll go into more detail in a moment. But think about diffusion. And you learned about diffusion last semester. Diffusion happens faster over short distances. So all of the hemoglobin molecules on the right side with the biconcave disc have a very short distance to travel to get from where they are inside the red blood cell to the outside. Whereas if you had a sphere-like cell, then the hemoglobin and the oxygen is going to have to travel much farther. If you look at the ones on the inside of the cell, that oxygen would have to diffuse way out here to get outside of the red blood cell. So that is why the large surface area to volume ratio is important, because it allows oxygen to quickly diffuse into and out of red blood cells. Another reason the structure is important is because of this biconcave disc structure. The red blood cells can form stacks, kind of like dinner plates, one on top of another, and these are called rouleaux. And it also allows the red blood cells to bend and flex to enter small capillaries. And both of these functions, again, are allowing the red blood cells to pass smoothly through the tiniest vessels. So here's a picture from your textbook. Some of these capillaries are very narrow. They're only the width of a single red blood cell. So the fact that the red blood cells can stack, like here's a rouleau right here, here's another rouleau over here, it allows them to pass through these very narrow vessels. And I actually have a video to show you of actual red blood cells passing through capillaries. So I'm not going to show you the whole 5-minute video, but I just want to show you some of it, because this will allow you to visualize how small these blood vessels are. So they have they've used a filter that makes the red blood cells black in this image. So up here at the top, you can see this would be like a venule. So this is a larger vessel, and you can see the blood cells are moving much faster. And then down here at the bottom are single capillaries where the red blood cells can only go through one at a time. So you see how they have to like squish and bend to get through these narrow capillaries. And I think we already missed a rouleau, but occasionally you can see them go through and like there was a stack right there. They can go through in like these stacks through these narrow capillaries. So this hopefully can help you visualize just how small these vessels are. Some of them are literally only big enough for one single red blood cell to squeeze through. So the structure of the red blood cell allows it to both have an efficient distribution of oxygen through the surface area to volume ratio, and allows it to smoothly pass through the smallest vessels in the body. So we'll continue on with the PowerPoint. So next we'll talk about hemoglobin, which can be abbreviated HB or HGB. So hemoglobin is making up 95% of all of the protein found in a red blood cell. So as I mentioned before, you can think of red blood cells as little hemoglobin packets, because that's pretty much all they are. So the structure of the hemoglobin looks like this. So this is a single hemoglobin molecule. They are made up of 2 alpha protein chains, which are shown in this picture as the darker blue. There's an alpha 1 chain and an alpha 2 chain. And then 2 beta protein chains, and they're shown in the lighter blue, beta 1 chain, beta 2 chain. And then each chain has a heme complex, which are these little discs here with the little red dot in the center. So because each chain has a heme complex, there are 4 hemes in total. And here is a close-up look at the molecular structure of a heme, and we're going to talk more about this in a moment. Note that fetal hemoglobin, which is the hemoglobin you have while you're in the womb, has a slightly different structure. Shortly after you're born, the fetal hemoglobin structure switches over to this one, which is the adult version. And the fetal hemoglobin has a higher affinity for oxygen, meaning it wants to hold on to oxygen more than the adult hemoglobin molecule, and this allows babies in the womb to basically steal oxygen from the mother through the placenta. If their hemoglobin had the same affinity for oxygen as the mother's hemoglobin, the baby's hemoglobin would never be able to get oxygen from the mother's hemoglobin. The mother's hemoglobin would hold on to it. And so for this reason, the fetal hemoglobin has a higher affinity for oxygen. So as promised, we're going to look a little bit closer at the heme. So again, just to remind you, we have a red blood cell or your erythrocyte, and this is made up of hemoglobin molecules. Hemoglobin molecules have 4 protein chains. Each protein chain has a heme unit on the inside. If we take a closer look at the heme unit, we can see the heme is this little disc right here in the middle. So all these ribbons around it are one of the protein chains. And this teal colored molecule here, these are 2 amino acids sticking off of the protein chains. So what we're looking at is this molecule right in the center, that is the heme. So the center of the heme, so the center of each heme is a single iron ion, which is abbreviated Fe2+. So this giant yellow circle in the middle, that would be the iron atom. And the iron atom can interact with a single oxygen molecule. So this little red molecule here with the 2 red balls with a double bond, that is an oxygen molecule, or O2. So when hemoglobin is bound to oxygen, it's called oxyhemoglobin, and it technically should be abbreviated HbO2. And when oxygen is bound to hemoglobin, it makes the blood a bright red color. Without oxygen, it should technically be deoxyhemoglobin, which is abbreviated HB, and this makes the blood dark red. However, because of the way light refraction happens, it can appear blue through light-colored skin, and for this reason, the early anatomists started drawing arteries as being red and veins as being blue, which still persists to today. So on all of the models in the lab and in the textbook, whenever you see a blue colored vessel, that's going to represent the deoxygenated blood, and the red vessels are going to represent the oxygenated blood. And that is based on the difference in the hemoglobin color with and without oxygen. So what is the function of hemoglobin? To put simply, it carries oxygen, and it can also carry carbon dioxide, but its primary function is going to be to carry oxygen. So one red blood cell has about 280 million hemoglobin molecules. Each hemoglobin has 4 hemes. Remember, 4 protein subunits, each one has a heme. So one hemoglobin can carry 4 oxygen molecules. So 280 million hemoglobins times 4 oxygen molecules each means that a single red blood cell can carry over 1 billion oxygen molecules. So stop and think about that for a minute, because you've got a lot more than just one red blood cell in your body. So oxygen can bind and unbind to hemoglobin based on the surrounding oxygen concentration. So when oxygen levels are high, like in the lungs, oxygen is going to go into the red blood cell and bind to hemoglobin. When oxygen levels are low, like out in the peripheral tissues, oxygen will leave the hemoglobin and diffuse into the low concentration area, which will be the interstitial fluid and the cells. And so in this way, the blood can carry oxygen from high concentration areas like the lungs to low concentration areas like our cells and tissues. Note that each heme molecule, that iron atom, can also bind to a molecule of carbon dioxide, forming carbaminohemoglobin. And we're going to get into more detail on all of these processes when we actually get to the respiratory system in chapter 23. We're going to take a quick segue to talk about the 3 types of anemia. Your textbook kind of scatters these throughout the different sections, but I wanted to bring them all together and talk about them at once. So anemia is defined as low oxygen delivery to the peripheral tissues. So basically, the cells of your body are not getting enough oxygen. This can be caused by a low hematocrit. And remember, the hematocrit is the percentage of formed elements in the blood, and 99.9% of the formed elements are red blood cells. So a low hematocrit would mean that you don't have enough red blood cells to carry the oxygen. It can also be caused by a reduced amount of hemoglobin inside the red blood cells. So you could have the normal number of red blood cells, but each red blood cell doesn't contain as much hemoglobin as it should. And anemia ultimately leads to a host of problems that start out minor and then can become more severe, because effectively your organ functions are deteriorating from a lack of oxygen. Remember, oxygen is necessary for the mitochondria to make ATP, and ATP is necessary for just about everything your body does. So common symptoms of anemia are fatigue and low energy, increased heart rate, shortness of breath, headaches, and dizziness. The fatigue and low energy is just your body not having enough ATP, essentially. The increased heart rate and shortness of breath is because your body is sensing a lack of oxygen. And so by increasing the heart rate and causing you to breathe faster, it's your body's way of trying to get more oxygen into the system. And then the headaches and dizziness are the effects of oxygen deprivation on the brain. The brain uses up a lot of oxygen, and the brain is one of the first organs that is affected by a lack of oxygen. There are three major types of anemia that we're going to talk about: sickle cell anemia, pernicious anemia, and iron deficiency anemia. These are by no means the only types of anemia, and I'm actually going to talk about another type of anemia in the in-class worksheet, but these are three of the major and most common types. So sickle cell anemia, also called sickle cell disease, is caused by a genetic mutation that alters the structure of the hemoglobin. The most common mutation is in that beta protein subunit. So basically the mutation causes the hemoglobin molecules to clump together and form these long chains inside the red blood cells. So the affected red blood cells end up to be long and skinny and sickle-shaped, hence the name, and not biconcave discs like they should be. And we talked about the importance of the structure of a red blood cell to its function. So when you change the shape of the red blood cell, like in sickle cell anemia, it can cause problems with blood flow. So the sickle cells can clump together and form clots. They also become sticky sickle cells, and they can stick together, and it makes the red blood cells not be able to pass through those narrow vessels as easily as the biconcave disc red blood cells. The red blood cells also only live for about 10 to 20 days, so they don't live for the full 120 days. And so often the process of making new red blood cells can't keep up with the red blood cells that are dying, and so that can cause these episodes of crises with the sickle cell disease. It also increases the stroke risk because there's an increased risk for clotting, and strokes are caused by clots that occur in the blood vessels in the brain. And it can cause episodes of severe pain called pain crises. Sickle cell anemia, the mutation, actually stuck around and became very common in our population because it actually helped to defend people against malaria. So it evolved in regions that were subject to malaria. So the incidence is higher in populations that are descended from ones that lived in areas that were subjected to malaria. Like if you have African, Middle Eastern, Mediterranean, Central or South American or Asian Indian descent, you may be more likely to get the sickle cell mutation. So if you have sickle cell disease, you have to have 2 copies of the mutation. So we all have 2 versions of each gene. One we got from our mother, one we got from our father. Both versions have to be mutated to have sickle cell disease. If you only have 1 copy, that is called sickle cell trait, and you're essentially a carrier for the sickle cell disease. So in the U.S., 100,000 people have sickle cell disease, but 2 million people have the sickle cell trait, meaning that one of their two genes has the mutation. And it is a big worldwide problem because 20 million people worldwide have sickle cell disease. Pernicious anemia is an anemia caused by a vitamin B12 deficiency. So vitamin B12 is necessary for red blood cell formation. So this could be caused by a lack of vitamin B12 in your diet, so you're just not eating enough vitamin B12 and you don't take any supplements. Or it could be a problem with a compound called intrinsic factor, which is a compound produced in your stomach that allows vitamin B12 to be absorbed. So if this is the cause of the anemia, you can't take a vitamin B12 supplement to counteract it because your problem is not being able to absorb the vitamin B12 in your digestive tract. So people with a problem with their intrinsic factor actually have to get daily vitamin B12 injections. So you're bypassing the digestive system entirely. And the last major type of anemia is iron deficiency anemia, which is anemia caused by a lack of iron in the diet. Iron is required for the heme formation. As we looked at before, each heme has an iron atom in the center of it. Each hemoglobin molecule has 4 hemes. Too much iron can also cause problems too, so don't just run out and take an iron supplement because it sounds like a good idea. Always consult your doctor before you start any iron supplements. And you can also get a lot of iron through your diet. So foods high in iron include red meat, fish, and most all meats have some high level of iron. Red meat is the highest. You can also get it from spinach and other greens like collard greens, turnip greens, etc. Beans, there's some fruits out there like strawberries and many more. So usually the people with iron deficiency anemia are like vegetarians that don't have a wide enough diet to supply them with enough iron, or people who are always eating unhealthy types of foods and not getting enough meat, vegetables, and beans in their diet. In this section, we're going to look at red blood cell production, which is called erythropoiesis, and we'll also look at the regulation of erythropoiesis. So, erythropoiesis is the process of red blood cell formation. This occurs in myeloid tissue, which is also known as red bone marrow. You can find the type of red bone marrow that makes new blood cells in the vertebrae, sternum, ribs, skull, both of your scapulae, hip bones, humerus, tibia, and femur. So the flat bones plus the epiphyses and metaphyses of some of your larger long bones. So the whole process starts with stem cells called hemocytoblasts. Hemocytoblasts are going to be the stem cells for all of the different types of blood cells, white blood cells, red blood cells, and platelets. Although for right now we're just focusing on the red blood cell pathway. These are also known as hematopoietic stem cells, or HSCs, and they're shown down here in the image. And again, we're looking at some red bone marrow or myeloid tissue, and in this case we're looking at the femur. So hemocytoblasts are going to divide to produce 2 types of stem cells. There are myeloid stem cells, and these are going to become red blood cells, platelets, and several types of white blood cells. And then there are lymphoid stem cells, which will only become one specific type of white blood cell called a lymphocyte. So the myeloid stem cell that is destined to become a red blood cell, called the progenitor cell, is going to differentiate into a cell called a proerythroblast. So up here on day 1 we have the proerythroblast. Then that cell is going to go through a series of erythroblast stages over the next 3 days. And during this process, it will be actively synthesizing tons of hemoglobin. Because remember, red blood cells are just little packets of hemoglobin. And so you don't need to know these separate stages of the erythroblast stage. We're just going to call these the erythroblast stages. You can tell the difference in what stage they're in under a microscope based on the way they look, which is based on how much hemoglobin they have made to that point. The final erythroblast stage is called the normoblast, and at this point the nucleus gets ejected from the cell. So most of the hemoglobin has been made. We have enough mRNA around to keep making some more hemoglobin, so we don't need the nucleus anymore, so the nucleus is ejected. After the nucleus is ejected, you have the reticulocyte. This is an immature red blood cell. It has about 80% of its final hemoglobin, and this is the cell that is actually going to leave the bone marrow and enter the bloodstream. So about 0.8% of all of the red blood cells in your circulation at any given point are going to be these reticulocytes. After about a day in the bloodstream, the reticulocyte matures into a fully developed red blood cell with all of the hemoglobin that it's going to have. So that is the process of erythropoiesis. Now we'll talk about how it's regulated. So erythropoietin, also known as EPO, is the hormone that directly stimulates erythropoiesis, which again is the red blood cell formation. So erythropoietin is secreted by both the kidneys and the liver. It's released when the kidneys sense low oxygen levels. So the kidney secretion of EPO is much more studied and understood than the liver secretion, and we know that under normal oxygen conditions, only very few cells in the kidney are secreting erythropoietin, but when you have a condition of low oxygen, which is called hypoxia, then you have an increase in the secretion of erythropoietin by the kidney cells. So again, hypoxia is a condition of low oxygen in the tissues. So the main triggers for erythropoietin secretion are anemia. So remember, anemia can be caused by a lack of red blood cells or a lack of hemoglobin in the red blood cells. When blood flow to the kidney declines, so remember the kidney is sensing low oxygen levels, so if you cut off blood flow to the kidney is going to therefore not have enough oxygen, and it's going to think the oxygen levels are low and secrete EPO. Also, if the oxygen content of air in the lungs declines, because this is going to cause the oxygen content of the blood to decline. And if you have damages to your respiratory surfaces in your lung, again, you're not going to get sufficient oxygen into the blood, which is going to cause low blood oxygen levels, which is going to be sensed by the kidney. So basically, all of these sections here, all three of these triggers, are causing the kidney to sense low oxygen levels. So when erythropoietin is released, it targets the red bone marrow or myeloid tissue. It has two main effects. It stimulates the cell division rates in those stem cells, so the hemocytoblasts and the myeloid stem cells, and it also stimulates cell division rates in the erythroblast in those erythroblast stages. It also speeds up the maturation of red blood cells by accelerating the hemoglobin synthesis. Athletes can try to enhance their performance by doing a process called blood doping. This is either taking erythropoietin, a synthetic form, or they can have their blood drawn, they can have the red blood cells spun down and frozen and saved, and then they inject or reinfused those red blood cells back into their bloodstream right before an athletic event. In both of these cases, taking the EPO or doing the reinfusion of red blood cells removed earlier can increase oxygen delivery to muscles by boosting red blood cell numbers. And remember, the function of a red blood cell is to carry around oxygen. However, this has bad side effects, because if you're increasing your red blood cell numbers, you are increasing your hematocrit. Remember, the hematocrit is the percentage of formed elements in the blood. So you're making your blood thicker, so more cells and less fluid. So it makes the blood more viscous or thick, which is going to increase the workload and strain on the heart. So you are potentially facing stroke, heart attack, and kidney damage from using practices such as blood doping. Other hormones can also affect erythropoiesis. So thyroxine, one of the thyroid hormones, androgens, and growth hormone can all increase erythropoiesis. We mentioned earlier that the effect of the androgens is why men have a higher hematocrit than women. And erythropoiesis also requires amino acids to make the protein, the hemoglobin protein. You also need iron and vitamins. So we talked a little bit before about vitamin B12. This is very important to the synthesis of the heme and hemoglobin. Vitamins B6 and folate or folic acid also play a role. And then sufficient dietary iron is also necessary, as we discussed earlier when we talked about anemia. So lack of vitamin B12 leads to pernicious anemia. Lack of dietary iron leads to iron deficiency anemia. So here is a summary of what we've talked about so far. So a low oxygen level in the blood is going to cause the kidneys to sense the low oxygen levels. The kidneys will then secrete erythropoietin. Erythropoietin will stimulate red blood cell production in the bone marrow. In order to make the new red blood cells, you're going to need things like iron and vitamin B12. And then the red blood cells are going to enter the general circulation, where they survive for about 120 days. In this section, we're going to look at red blood cell turnover and recycling. So remember that red blood cells have a very short lifespan relative to the other cells in our body. And here are some more astonishing numbers for you. You have about 25 trillion red blood cells circulating in your blood right now. That would be the number 25 followed by 12 zeros. They have a rough life, lots of physical stresses being bumped around inside all of those vessels. So a typical red blood cell only lives for about 120 days. So you're constantly having to make new red blood cells. So about 1% of your circulating red blood cells are replaced every day. And while 1% doesn't sound like a lot, 1% of 25 trillion is still 250 billion. So you are still making 250 billion new red blood cells every day, which, if you do the math, comes out to be about 3 million new red blood cells coming into your bloodstream every second. So this always blows my mind when I look at these numbers. So every second, 3 million new red blood cells are entering your bloodstream from the red bone marrow. So think about how many million were added to your bloodstream just since you started looking at this slide. So all of this begs the question, what is happening to the 250 billion old worn out red blood cells that we're replacing every day? So if we're making 250 billion new ones a day, something has to be done with the 250 billion worn out ones that we're replacing. And that's what the focus of this section is about. So the old red blood cells are phagocytized by these large white blood cells called macrophages. So the macrophages use phagocytosis, which is a type of endocytosis, to engulf the old worn out red blood cells. Here's a picture showing you the comparison of the sizes. So this is a large macrophage right here in the center of this picture, and then these are a bunch of red blood cells around it. So you can see that macrophages are much larger than red blood cells, so they are able to engulf and phagocytize individual red blood cells. Here's another cool image that actually shows a single macrophage engulfing a single red blood cell, also called a erythrocyte. So he's coming up to it here, he's starting to engulf it here, and he's just about completely engulfed it down here. The macrophages can also detect debris from red blood cells that have ruptured and burst open, which is called hemolysis, and they can phagocytize the debris as well, so like the bits of hemoglobin that get released. This process usually occurs in the spleen, liver, and red bone marrow. So these are three areas where the macrophages are specifically monitoring the red blood cells to see if they are still okay or if they're getting old and worn out and it's time to take them out of circulation. So just a little segue into what happens if you have an abnormal amount of hemolysis. So normally the macrophages engulf the red blood cells before they degrade to the point that they burst open. And when they do degrade to the point that they burst open, this is called hemolysis. So this is destruction of a red blood cell. The red blood cell will burst open and the hemoglobin is released to just float around inside of the blood vessels. And if this happens too quickly, and if you have a bunch of red blood cells that are suddenly bursting open and dying, the macrophages can't recycle all of the hemoglobin quickly enough, and hemoglobin ends up getting released into the urine, and this is called hemoglobinuria. And this is when you have a brown or red colored urine from the presence of hemoglobin. And again, this is caused by an abnormally large number of red blood cells breaking down. And this is not to be confused with a different condition called hematuria, which is the presence of intact red blood cells in the urine. So this would what you would call blood in the urine. And this occurs with kidney damage or damage of vessels in the urinary tract, so you actually have active bleeding into the urinary tracts, so the red blood cells in this case would still be intact, whereas with hemoglobinuria, this is just the hemoglobin because the red blood cells have broken down. So back to the macrophages and the recycling of the red blood cells. So I put a picture of a hemoglobin up here to remind us of the different parts. So remember, we have the 4 protein chains, we have 2 alphas and 2 betas. We also have the heme groups, and in the middle of the heme groups we have an iron atom. So after the red blood cell is engulfed by the macrophage, first those protein chains are separated from the heme groups. So the heme groups are stripped away from the protein chains. The protein chains are broken down into their individual amino acids. Those amino acids can then be used to build new proteins, either new hemoglobin chains or other proteins the body needs. Second, the iron ion is stripped away from the heme group. The iron is recycled, so it's transported through the blood using a transport protein called transferrin. And so the name tells you it's transporting iron, because iron, the Latin name for iron, is ferrous. So it's transported back to the red bone marrow. And so the developing erythroblasts in the red bone marrow that are undergoing that process of erythropoiesis we looked at in the previous section can use this iron to make new heme groups in order to make new hemoglobin. So the iron recycling process is actually very efficient. So again, remember, you're making 250 billion new red blood cells every day. And so to make all of those new red blood cells, you need 26 milligrams of iron per day to make the new hemoglobin. But you only need 1 to 2 milligrams of dietary iron per day to meet this need. The rest of that 26 milligrams comes from recycled iron from the red blood cells that are being broken down and recycled. So very, very efficient process. You're only needing to replace a little bit of that iron every day through your diet. All right, so we've talked about the protein chains, we've talked about the iron, so the last thing is the heme group. So the heme group must also be broken down, and this is after we've taken the iron away from it. So after the iron is removed, heme is converted to a molecule called biliverdin, and biliverdin has a greenish color to it, and I'll tell you why that's important in a second. The biliverdin is then converted to bilirubin, and bilirubin has an orange-yellow color to it. And then the macrophages at this point are going to release the bilirubin into the bloodstream. And then on the next slide, we're going to follow what happens to the bilirubin. But for now, I want to point out that this recycling process that takes place inside the macrophages is why your bruises change color over time. So when you first get a bruise and it's very fresh, it may appear pink or red because the hemoglobin that is released from the blood vessels, and so let me back up, a bruise occurs when blood vessels underneath the skin break, and so blood is released into the tissues, and that's what causes a bruise. So at the very beginning of a bruise, the hemoglobin is still carrying oxygen and it has a pink or bright red color. It very quickly will start to fade to a blue or purpley color. This is because the hemoglobin has now lost its oxygen, so it has that darker color to it. And then the macrophages are going to come into the tissue and start cleaning up. And as the macrophages are cleaning up all of those red blood cells that have gotten out of the blood vessels, the macrophages are going to start breaking down those heme groups into biliverdin, and your bruise is going to start taking on a greenish appearance. And then a few more days, that biliverdin is converted into bilirubin, and your bruises take on a yellowish color. So this is why your bruises change color over time. And sometimes when you look at a single bruise, you can see all the different colors in one big giant bruise, and that's because different areas are healing at different rates. Maybe some of the areas the macrophages were able to get there more quickly than in other areas, and so they started the cleanup process earlier. So what happens to the bilirubin that we left in the bloodstream? So we said that the macrophages then dump the bilirubin into the bloodstream, and so this bilirubin is then filtered out by the liver. So jaundice is a condition where you have a yellowish tint to your skin and the sclera of your eyes, so the white of your eyes, due to a buildup of bilirubin in the body. And jaundice can happen whenever you have a liver problem that prevents the liver from filtering the bilirubin out of the blood. And so you can see jaundice in a number of different conditions that affect the liver, like liver cancer, liver failure, with hepatitis, even a gallbladder problem. If your gallbladder gets blocked by gallstones, that can cause jaundice. Anything that's basically backing up this process from occurring in the liver. So the liver puts the bilirubin into the bile it produces, and we're going to talk about this production process later this semester when we get to chapter 24. We'll talk about the liver and how it makes bile. Bile is actually made by the liver, not the gallbladder. And then the bile that the liver makes goes into the small intestines, and it's now got the bilirubin in it. So then that bile and the bilirubin is going to go through the small intestines to the large intestines. And in the large intestines, bacteria convert bilirubin to two different types of molecules. Some of the bacteria convert it to stercobilins, which have a brown color, and some of the bacteria convert it to urobilins, which have a yellow color. And you may have never wondered this, but if you ever did, this is why your feces are the color that they are. So your feces are usually anywhere from a brownish to yellow color. If they're more brown, then you've got more stercobilins. If they have more of a yellowy color to them, you've got more of the urobilins. And so again, you are breaking down and recycling 250 billion red blood cells every day. So that's why you have enough of these compounds in your feces to actually change the color. So finally, here is a summary of the whole red blood cell turnover process. So everything that I just discussed on the previous slides are shown in this picture. So if you start up here in the top left corner, this is the macrophage. And again, the macrophages in the spleen, liver, and red blood, bone marrow are the ones primarily doing this. So they take in the old worn out red blood cells. They also can take in the pieces of red blood cells that have ruptured and undergone hemolysis. Then they're going to break those red blood cells down into the amino acids, which can be recycled and used to make new protein. The heme group is going to have the iron removed. Iron is going to be transported back to the bone marrow by transferrin, again to make new red blood cells. The heme group without the iron is converted to biliverdin. Biliverdin is converted to bilirubin. Bilirubin is put out into the bloodstream. It's then filtered out by the liver. The liver excretes it in the bile. It makes its way to the large intestines where it's converted into urobilins and stercobilins. Some of the urobilins can be reabsorbed into the bloodstream where they are secreted by the kidneys, and this is actually why your urine can have a yellow color, because remember, urobilins has a yellow color. So the urobilins can also be excreted in urine. And then the other parts of this graph is just showing you that if you have too many red blood cells that undergo hemolysis all at once, instead of the macrophages being able to recycle it all, sometimes it can get directly released through the kidney, but that is called hemoglobin urea, and that's not a good thing. And then up here is just showing you the production of new red blood cells. So this is everything we just talked about in this section. In this section, we're going to talk about blood typing. So first, a little background information. So if you think back to last semester in chapter 3, we talked about the functions that membrane proteins can have. And one of those functions was that membrane proteins, and usually ones called glycoproteins, acted as recognition molecules, so like little ID tags for the cell. So to refresh your memory, you have these proteins that can be embedded in the cell membrane, and with a glycoprotein, you've got the little sugar molecules that can stick out and face the outside of the cell, and these can often act as ID tags to identify a cell to other cells in the body. So now we're going to have a little preview of some information from chapter 22, the immune system. So we use the term antigen to describe any substance that can potentially trigger an immune response. So these little ID glycoproteins on our cell surfaces are types of antigens. And this is how our body recognizes cells that belong to us, so our self cells versus a foreign cell. So if an antibody for a specific antigen sees that antigen on a cell, it's going to say, "Oh, you don't belong here," and it's going to attack it as if it were foreign. We do not have antibodies for antigens we have. That's how our bodies can tell self cells from foreign cells. So antibodies are immune system molecules that attack foreign antigens. So when antibodies attack a foreign antigen, it causes clumping, which is also called agglutination. So, for example, and we'll get to this level of detail in a moment, your red blood cells do have surface antigens, or I should say they can have surface antigens, which are these little glycoproteins that stick out from the cell surface. If there's an antibody that recognizes that antigen, it's going to see it as being foreign, and it's going to attack it, which is going to cause this agglutination or clumping, which is then going to lead to hemolysis, which is when the blood cells erupt and basically die and spill all their hemoglobin out. And antibodies are very specific for the type of antigen they attack. So there's probably been a lot of talk about antibodies in the news in the last couple of years because of COVID. So we do develop antibodies for bacteria and viruses as well, not just for these ID tags. And so you've probably heard in the news about like people having COVID antibodies in their blood. So when I'm talking about antibodies being very specific, if you have antibodies for COVID, those antibodies are not going to recognize the flu virus, and vice versa. If you have flu antibodies, they're not going to recognize the COVID virus. So all antibodies are very specific. So this antibody here that recognizes this specific antigen is only going to recognize that specific antigen. All right, so let's get to red blood cells. So all cells in your body contain surface antigens, every single one of them. But in this chapter, we're focused on blood and red blood cells. And red blood cells have three very important surface antigens. They are called A, B, and Rh factor. All right, so these are the three important surface antigens found on red blood cells. So again, an important note to remember and keep in the back of your mind, your blood will not have antibodies against antigens found on your cells, but you will have antibodies for antigens you don't have, right? And this maybe you'll make a little bit more sense in a second when we look at these pictures. So if you have type A blood, then your red blood cells have a surface antigen A on them. It's a specific type of antigen. You will not have antibodies that recognize antigen A, because otherwise they would attack your own blood cells and you would die. However, you may also have in your blood antibodies that will attack the B antigen, because you don't have the B antigen, you only have the A antigen. Let's look at another example. If you have type B blood, your red blood cells have the type B antigen on their surface. So you're not going to have anti-B antibodies, because again, that would attack your own blood and you would die. But you will have anti-A antibodies because you don't have the A antigen on your blood cells. If you are type AB, then you're going to have both A and B antigens on your blood cells. So notice you've got the little red circles for A, the little blue triangles for B. You've got both of them. And if you have type AB blood, you're not going to have either anti-A or anti-B antibodies, because again, if you had those antibodies, they would attack your own blood cells. So again, remember, the antibodies are there to attack foreign things. So the antibodies are only going to match things that you don't have in your body. And then if you have type O, O actually is like O0 for nothing there. So type O blood has red blood cells that don't have either A or B antigens. So no A, no B antigen. And because you don't have either of the antigens, you would have both types of antibodies. You would have anti-A antibodies and anti-B antibodies. Then your textbook didn't really point out what was going on with RH, so I tried to make my own little picture here for you. If you are RH positive, so if you have a positive after your blood type, for example, if you're like B positive or A positive, so if you have that little positive, what that means is that your red blood cells have the RH factor antigen on their surface. And I drew a little green square to represent the RH antigen. So you would have the RH antigen on your blood cells, so you would not have anti-RH antibodies in your plasma. And if you are Rh negative, so you have the little negative after your letter, then your your red blood cells are not going to have the Rh antigen, but you could have anti-Rh antibodies. Now, the anti-Rh antibodies are a little bit different than the A and B antibodies. With the A and B antibodies, you're pretty much born with those. So if you're type O blood, you have anti-A and anti-B antibodies from the time you're born. But if you are Rh negative, you don't get the anti-Rh antibodies until you're exposed to the Rh antigen first at some point in your life. And so you have to be what we call sensitized to it before you get these antibodies. But you still have the capability to get these antibodies, whereas if you're Rh positive, you would never get the antibodies because otherwise they would attack your own blood cells. All right, so now let's look at blood typing. So to determine a person's blood type, like if you don't know what your blood type is, we take your blood, a little sample of your blood, it just takes a little drop from a finger, and we expose it to all three types of antibodies. So remember, anti-A antibodies are going to bind to A antigens. Anti-B antibodies are going to bind to B antigens. Anti-Rh antibodies will bind to the Rh antigens. All right, so the little card or the little vial or however they test the blood is coated with these antibodies. And then what we do is we look for clumping. In a blood typing test, the clumping determines the presence of those antigens in the blood you are testing. So let's look at an example. So each of these rows is a different person's blood. So let's just look at the first row. So at the first row, we put a person's blood in these three spots on the card. There's a spot for anti-A, a spot for anti-B, and a spot for anti-Rh. We see clumping in the anti-A spot, no clumping in the anti-B spot, and clumping in the Rh spot. That means this person's blood has the A antigen, and it also has the Rh antigen. So that's what caused the clumping, was because their blood had those antigens. So this person would be a type A positive. They have the A antigen, they have the Rh antigen. In this next row, for a different person, you know clumping in the anti-A, clumping in the anti-B, clumping in the anti-Rh. That means they have the B antigen, they have the Rh antigen, so they would be B positive blood type. If you get clumping in all three spots, then that means you have all three antigens. So that would be type AB positive. You have the A, you have the B, and you have the Rh. And then in this last example, if there's no clumping in any of the three spots, then you have no antigens, no A, no B, no Rh. That would be type O. The O says no A and no B, and negative says no Rh. You can also go the other way for practice. Like if you want to practice your understanding of this, stop and think for a minute, where would you see clumping if you had type A negative blood? Well, A negative blood tells me that I have the A antigen, and that's it. I don't have the B antigen, and the negative means I don't have the RH antigen, so therefore I should only see clumping in the anti-A place on the blood typing card. All right, if I have B negative, again, this is telling me I only have the B antigen. I don't have an A and I don't have an RH because I have a negative, so I should only see clumping in the anti-B column. And then with AB negative, I should expect to see I have an A antigen and I have a B, but I don't have the RH, so I should have clumping with the A, clumping with the B, no clumping with the anti-RH. And then think about O positive. So remember, O is telling me I don't have A or I don't have B, but the positive is telling me I do have the RH. So I would expect to see no clumping in A, no clumping in B, but I do get clumping in the anti-RH column. So again, if you need to practice this some more and trying to figure out what people's blood types are based on the different clumpings and then who can get what type of blood, there's an excellent little blood typing game, and I'll post a link to it on D2L. Play it as many times as you like. It actually can be a little fun, and it can give you some great practice. So blood typing, why does it matter? Well, a transfusion is the process of transferring whole blood or plasma from one person to another. And if you don't transfuse the right type of blood, you can get a cross reaction. A cross reaction occurs if the recipient, the person getting the blood, has antibodies for antigens on the donor's red blood cells. This can be quickly life-threatening because the recipient's antibodies are going to wage war on that donated blood, and you'll get massive hemolysis of those red blood cells, and then all the little pieces of the blown up red blood cells can cause blood clots, and it can be very life-threatening. So for that reason, oh, and here's a picture that shows, for example, if you have a donor that is giving you blood and the donor has type A blood, but the person you're giving the blood to, like in this case, this is a type B person, well, the type B person is going to have anti-A antibodies in their body. So as soon as you give them the type A blood, their antibodies are going to attack the red blood cells and cause this potentially fatal reaction. And it's most important to consider the donor's antigens, so what antigens are on the surface of the donor's red blood cells, and then what antibodies the recipient has. This is the reaction you have to worry about the most. The opposite reaction between a donor's antibodies and the recipient's antigens is very minor due to the volume, the two different volumes of blood. So this is what we're considering, the donor's antigens and the recipient's antibodies. So because we don't want to have a cross reaction, prior to giving a blood transfusion, there are two steps that they follow. First, they determine the blood type of the patient that needs the blood. And then once they've determined that blood type, they do a second step. They do a cross-match test before actually giving the patient donor blood. And with cross-match testing, they take the donor's red blood cells and they expose it to a sample of the recipient's plasma under controlled conditions. And this is just to make sure that there's no issues with other surface antigens. So A, B, and RH are the three major surface antigens, but there are other smaller minor ones out there. And so the cross-match testing just makes sure that there isn't a reaction, even if, say, both the donor and the recipient have the same blood type. In emergencies, however, there isn't always time to do blood typing and cross-match testing. So for that reason, we talk about the universal donor, and there used to be more emphasis on the universal recipient, not so much nowadays, but I'll still explain it to you to help you understand these concepts. So with the universal donor, this is going to be type O negative blood. So why are they the universal donor? Well, remember, let's go back a slide. The most important thing to consider, donors, antigens, recipients, antibodies. So the donor's antigens are important to consider. Well, with the type O negative blood, you've got no surface antigens, right? So there's no surface antigens for the recipient's antibodies to attack. So remember, type O negative has no A antigens, no B antigens, and no RH surface antigens. So no A, no B, no RH. That means you can give type O negative to anybody and they won't have a reaction. However, if you have type O blood, if you have type O negative blood, you can only get blood from another type O negative because you have all of the antibodies in your plasma. On the flip side is the universal recipient. So this is a person who can receive any type of blood. The universal recipient would be a type AB positive. And remember, for recipients, it's the antibodies that matter the most. And with type AB positive, you have no plasma antibodies to attack a donor's antigens. And we have no plasma antibodies because we have all the antigens. We have the A antigens, we have the B antigens, and we have the RH antigens. So I'm not going to have any antibodies that can attack my own blood cells. So I have no anti-A antibodies, no anti-B antibodies, and no anti-RH antibodies. So it doesn't matter whose blood I get, I've got nothing to attack it. All right, so the universal donor is going to be the one with no antigens. The universal recipient is going to be the one with no antibodies. So again, that goes back to the most important thing to consider is the donor's antigens compared to the recipient's antibodies. And because O negative is the universal donor, quite frequently in medical dramas and TV shows, when there's an emergency and a patient's being rushed into the operating room, you can hear them say things like, have 6 units of O neg on standby, right? And so they're actually talking about having O negative blood ready to go because it's an emergency and they don't have time to do blood typing and cross-match testing, so they will just use O negative. And then one last part while we're on blood typing, anti-RH antibodies can cross the placenta. So this can cause problems if you have an RH negative mother who has an RH positive baby. So during the first pregnancy, she can be sensitized to the RH antigen, and this occurs because during the pregnancy, the mother has no antibodies yet, because remember, the anti-RH antibodies don't develop until you're sensitized, but the fetus has these RH antigens on their blood cells. Well, during birth, right, birth can be a somewhat violent affair, and so you can get mixing of the blood. You can get hemorrhaging where the mother's blood supply is exposed to the fetus's blood supply. Now the mother's blood can see those RH antigens, and she'll start developing those anti-RH antibodies. So after that first birth, she now has these anti-RH antibodies, and that's shown by the little green antibodies here. Then, during a second pregnancy, those anti-RH antibodies can then attack the fetus's blood cells and cause what's called the hemolytic disease of the newborn. And so, again, during a second pregnancy, now she's got these anti-RH antibodies. They can cross the placenta and go in and attack the fetus's red blood cells, which of course is not good and can lead to death of the fetus. So this has been largely prevented nowadays because they do have shots that they can give RH negative mothers. RhoGAM is one common one, and this shot prevents them from making anti-RH antibodies so that you never get this whole process started. And that is a section on blood typing. In this section, we're going to look at white blood cells, which are also called leukocytes. Leuko means white in Latin. Cyte, again, means cell. And we're going to do an introduction, characteristics and functions, and some disorder terminology in this section. So white blood cells are more like a standard cell. They have a nucleus, they have all of their organelles, so they are not as specialized as red blood cells, although they do have their specialties. They also do not contain any hemoglobin. And there is only a small amount of white blood cells in the circulating bloodstream. So if you remember from earlier, less than 0.1% of the formed elements of whole blood are actually white blood cells. However, that's not to say that you don't have a lot of white blood cells. It's just that most of them are outside of the blood vessels in your connective tissues and in your lymphatic organs. So there is actually only very few that are in circulation at any given time. And so these white blood cells, they use the bloodstream as a means of transportation to get from one place to another place within the body, and also to get to the site of an infection or an injury. There are 5 types of white blood cells, and we'll go into more details in the next section, but there are basophils, eosinophils, neutrophils, monocytes, and lymphocytes. So the circulating white blood cells have 4 functional characteristics. All of them can migrate out of the bloodstream. So first they go through a process called margination, in which they slow down and they actually contact and adhere to the endothelial cells that make up the blood vessel walls. And then they go through immigration, which is when they squeeze between adjacent endothelial cells to enter the surrounding tissue. I show a video to my 2251 students to show them what it looks like inside of a cell when the cell's doing its jobs. But if any of you guys remember that video from a previous semester, it also showed a white blood cell that was slowing down and adhering to the endothelial cells of a blood vessel and then squeezing between them. And we're going to see another video in this lecture in just a couple of slides. So circulating white blood cells are also capable of amoeboid movement, and what that means is they can move like an amoeba, and that means they can stick out these little, they're called pseudopodia or false feet, so little cytoplasmic extensions that they can stick out, and then it allows them to sort of move in that direction. So all circulating white blood cells have this capability, and this is actually how they can exit the blood vessel, get back into the blood vessel, and how they can then go out and find where the site of injury or infection is. They are all also attracted to specific chemical stimuli, and this is called positive chemotaxis, when a cell follows a chemical trail and moves toward the source of the chemicals. And in this case, the chemicals are released by cells at the site of an injury or an infection, and basically those chemicals are like flags flagging down white blood cells saying, over here, you've got a problem over here, and then the white blood cells follow that chemical trail to get to where they need to be. Some of the white blood cells are capable of phagocytosis. Not all of them, but some of them are. And these are called phagocytes, is the general name for any cell that can undergo phagocytosis is a phagocyte. And phagocytes can engulf pathogens, cell debris, and other materials by using phagocytosis. So neutrophils and eosinophils are what we would call microphages because they're phagocytes, but they're relatively small cells. And then your monocytes are what we call your macrophages because they are much larger cells. So some white blood cell related terminology. A differential count is an estimate of the number of each type of white blood cell from a blood smear. So it's very similar to the slides of like blood cells that you looked at in lab in AMP1, where you can actually count the number of white blood cells amongst the different red blood cells, and you can differentiate which type is which. Leukopenia is a low or inadequate number of white blood cells. Leukocytosis is when you have an excessive number of white blood cells. This can occur during a routine infection. So if you are fighting off some type of infection, it could be bacterial, viral, fungal, a parasite, any kind of an infection, and you will have a temporary increase in your number of circulating white blood cells. However, if it's a very excessive increase, it's an extreme leukocytosis, that could be a sign that you might have leukemia, which is an actual cancer of the white blood cells. And because if you remember from AMP1, when we did talk a little bit about cancer, we talked about how cancer is uncontrolled cell growth. And so with leukemia, you get uncontrolled growth of white blood cells to the point where they even start squeezing out the red blood cells, and you end up with way too many around. That's going to hurt your oxygen carrying capability. And then these white blood cells are usually not functional because once they turn cancerous, they usually stop doing their normal jobs. In this section, we're going to look more specifically at the different types of white blood cells. So these are going to be your neutrophils, eosinophils, basophils, monocytes, and lymphocytes. All of these are leukocytes. So again, remember, white blood cell can also be called a leukocyte. And originally, the white blood cells were divided into two groups based on how they stained when you used a particular type of stain in the laboratory. So they divided them into granulocytes, which when you stain these cells, you could see granules in the cells. So that's how they got the name granulocytes. And these would be your basophil, eosinophil, and neutrophil. And then the other two, the monocyte and the lymphocyte, got called agranulocytes because when you stain them, you couldn't see granules. Well, now, it's later, we have more technology. We now know that those granules that were showing up in the staining were actually little lysosomes full of digestive enzymes. And the agranulocytes have them too. They're just smaller, so they're not as easily visible under a light microscope. So they got called agranulocytes to make it sound like they don't have the granules, but they actually do. So that's sort of an old way of classifying these, and it can still be helpful when you're trying to identify them under a microscope. But a better way to think of them is terms of their function according to the type of defense they offer the body. So your basophils, eosinophils, neutrophils, and monocytes are all part of what we call our non-specific defenses. This means they do not discriminate between one type of threat or another. They're going to react to most types of threats. Whereas your specific defenses, which are going to be your lymphocytes, will only respond to very specific threats. So for example, a lymphocyte is a type of cell that makes antibodies, and we already mentioned earlier in this lecture about how antibodies are very specific for what they attack. So an antibody for one thing is only going to attack that one thing. And we'll get a lot more detail in chapter 22 on the difference between specific and non-specific defenses. But for now, let's look at these 5 types of white blood cells. So we start with a neutrophil. Neutrophils got their name because they were hard to stain with acidic or basic dyes. So they were considered neutral, and that's how they got the name neutrophil. These are the majority of the white blood cells in your bloodstream. So 50 to 70% of circulating white blood cells are going to be neutrophils. So you can think neutrophil is the most numerous. Neutrophils can also be called polymorphonuclear leukocytes, and that is because the nucleus inside them looks like a bunch of beads on a string that are strung together. It can be anywhere from 2 to 5 of these little beads in a row. So this is another name you might see for a neutrophil. Neutrophils are highly mobile, so they're usually the first to arrive to a site of injury or infection. And they specialize in attacking and digesting marked bacteria. So neutrophils are phagocytes, and they can engulf invading bacteria. So I'm going to stop and show you a quick little video of someone who actually caught a neutrophil chasing down a bacteria.
This classic movie was made by David Rogers at Vanderbilt University in the 1950s. It shows a neutrophil, a type of white blood cell, chasing a bacterium through a field of red blood cells in a blood smear. After pursuing the bacterium around several red blood cells, the neutrophil finally catches up to and engulfs its prey. In the human body, these cells are an important first line of defense against bacterial infection. The speed of rapid movements, such as cell crawling, can be most easily measured by the method of direct observation.
And now he's going off to find another bacterium. So, neutrophils also have a pretty short lifespan. They only spend about 10 hours in the bloodstream, and then when they're actively engulfing pathogens and debris, like we just saw in that video, they might only live 30 minutes or less. And that make more sense to you on the next slide when we talk about what they do after they catch the bacteria. All right, so how neutrophils function. So as we saw in that cute little video, a neutrophil was able to chase this bacterium and then he engulfed it through phagocytosis. So then the next step is that the neutrophil is going to increase its metabolic rate. This is called a respiratory burst. And what this is going to do is increase a bunch of highly reactive molecules inside the neutrophil. Things like hydrogen peroxide and superoxide anions. So these things are dangerous, and they will help to kill the bacteria that has been engulfed. The vesicle that is holding the bacterium is also going to fuse with lysosomes within the neutrophil, and those lysosomes are going to contain defensins, which are proteins that can punch holes in the pathogen, which would be a bacteria in this case, and they also have digestive enzymes to break down the pathogen and or bacteria. So here is a picture of a neutrophil, and again, don't worry about getting bogged down into all the specific little details. What I just wanted to show you with this picture, where you can see the nice nucleus that looks like beads on a string. You can also see a microbe here that is being engulfed. You can see the production of those highly reactive molecules like hydrogen peroxide and superoxide anions, and then you can also see a lysosome here that has the digestive enzymes and defensins that are being released into the little cavity that has the microbe trapped. Neutrophils will also release prostaglandins and leukotrienes. Prostaglandins are compounds that will increase capillary permeability. This is as part of the inflammatory process. So by increasing the permeability of the capillary, more fluid is going to leave the bloodstream and go into the tissue, and that causes swelling. The leukotrienes are going to be chemicals to attract other phagocytes and say, hey, we have a problem here. I need your help to come clean it up. And neutrophils can only eat about 1 to 2 dozen bacteria before dying. That's why they have a short lifespan. And the reason that they end up dying so quickly after eating the bacteria is because these highly reactive molecules that they make to help them actually kill the bacteria are actually damaging to the neutrophil itself. So just the act of catching these microbes and killing them this way is also damaging the neutrophil itself. So it can only eat 1 or 2 dozen bacteria and then it dies, and that's why it has to call other cells to the area to help it fight the infection. And I know a lot of us really hate the idea of pus, looking at it, thinking about it, maybe even talking about it, but pus is a mixture of dead neutrophils, cellular debris, and other waste from an infection. So the next time you see some pus somewhere on your body, instead of being grossed out, think that it's full of all of these neutrophils who sacrifice their life in defense of your body. And now I have another video to show you that will show you several of the concepts that we've just talked about.
The white blood cells are an important part of our body's immune system. Neutrophils are a special group of white blood cells that play an extremely important role in protecting the body against infections. Mature neutrophils do not divide. New neutrophils are constantly formed in the bone marrow where they develop and mature. Mature neutrophils are released into the blood where they circulate for 3 to 12 hours and then move to other tissues where they survive only 2 to 3 days. These mature neutrophils can be found in large and small blood vessels, as well as in the tiny capillaries in different tissues. In the bloodstream, neutrophils act as surveillance cells searching for infections. Neutrophils can sense a site of infection because of chemicals that are released by the bacteria. Upon detecting these chemicals, neutrophils slow down and begin to stick to the walls of the blood vessel. They then squeeze between the cells of the blood vessel wall and enter the infected tissue. Here, the neutrophils crawl towards the infection as they continue following the chemical signals. At the site of infection, bacteria are coated with antibodies of the immune system and marked for ingestion. The neutrophil recognizes the marked bacterium, engulfs it into its body, and traps the bacterium in a sac called a phagosome. The neutrophil contains pouches called lysosomes, which are full of digestive enzymes and chemicals that can kill bacteria. The pouches, or lysosomes, then combine with the phagosomes containing the bacteria and release the enzymes and chemicals, resulting in the ultimate death of the bacterium inside the neutrophil. After killing and digesting the bacteria, the neutrophil is spent. Having completed its duty, the neutrophil shrinks in size and breaks up into smaller pieces that can be recognized and eaten by other cells.
So that was the neutrophils. The next type of white blood cell is the eosinophils. These stain red with a red dye called eosin, which is how they get the name eosinophils. They can also be called acidophils because they stain with acid dyes in general. These make up 2 to 4% of the circulating white blood cells. They typically have a bilobed nucleus, so a nucleus with 2 lobes. And they can also attack objects coated with antibodies, and they do have the ability to be phagocytes, but their strength lies in the fact that they can also attack via exocytosis of toxins. This makes them particularly effective against parasites that are too large to be engulfed. So eosinophils can surround the parasites, release the toxins, and that will kill the parasite. So a large number of eosinophils in a blood analysis can indicate someone has a parasite infection. Eosinophil counts also increase during allergic reactions, and they are also attracted to injury sites. However, whereas the neutrophils were releasing inflammatory chemicals, the eosinophils release anti-inflammatory enzymes to help counteract the pro-inflammatory actions of the neutrophils, basophils, and mast cells. And so the idea of this is to prevent the inflammation from like just running away and not being able to be stopped. So the eosinophil is kind of putting the brakes on the inflammatory response. So here is a picture. So if you have like a large parasitic worm, like a hookworm or a tapeworm, it's too large to be engulfed by a white blood cell. So the eosinophils can come right up to it, and they can release these cytotoxic chemicals from the inside, which can kill this parasite in the body. Here's another picture to give you another example. This is an immature fluke, which is a type of parasite, and you can see that it is surrounded by eosinophils. And I also have a video for this one as well. It's very short. So this is a very short video. So before I hit play, because it's only 11 seconds, what you're looking at here, these are a bunch of eosinophils, and this is a parasitic worm that has gotten into someone's body. And so we're going to see how the eosinophils are going to attack it. So you can see them moving around now. They're attacking, so they're all getting close. They're swamping that worm, and they're releasing all those cytotoxic chemicals. And we can watch it one more time. That worm is gone. Okay, so that was the eosinophils. Next we have the basophils. They are so named because they stain with basic dyes, so they got called basophils. They are small and very rare in the circulating blood, so there is less than 1% of all of the circulating white blood cells are basophils. Basophils migrate to injury sites, and once there, they discharge granules that contain both histamine and heparin. Both of these are pro-inflammatory molecules that will increase local inflammation at the site of an injury or infection. Histamine dilates blood vessels, which brings more blood to the area, which can contribute to swelling and the redness and the heat that you might feel in an inflamed area. And heparin prevents blood clots, and we're going to talk about blood clots specifically later. But again, by preventing the blood clots, it's allowing more blood to come to that area to help promote the inflammation. Note, if you remember from A&P 1, way back in chapter 4, when we talked about tissues and we talked about connective tissue proper, and we mentioned mast cells and how mast cells can release things like histamine and heparin as part of the inflammatory reaction. Well, the compounds released by the basophils are the same compounds released by the mast cells in the connective tissue, but keep in mind that basophils and mast cells are not the same. They're two distinct, separate types of cells. Basophils can also release chemicals to attract other basophils to the area, as well as eosinophils. And then that brings us to monocytes/macrophages. So these make up 2 to 8% of the circulating white blood cells. These are very large cells with a large nucleus. So look at the size of this cell compared to the red blood cells next to it. Monocytes only circulate for about 24 hours and then they leave the bloodstream to go out into the tissues. When they leave the bloodstream, they are then known as macrophages. So like earlier in this chapter, we were talking about how macrophages will engulf old and worn out red blood cells. Well, because they're not in the blood system at the time, they're called macrophages. So they're only monocytes for that about 1 day period that they're in the bloodstream, and then when they leave, we then switch and start calling them macrophages. Macrophages/monocytes are aggressive phagocytes. They will even try to engulf something as large or larger than themselves. They also can secrete chemicals to attract neutrophils, other monocytes, and fibroblasts to an area. And again, if you remember from AMP1, a fibroblast is a type of cell found in connective tissue proper, and fibroblasts can produce scar tissue. So they start making those collagen fibers to produce scar tissue to wall off an injured area. And then finally, we have the lymphocytes. These make up 20 to 40% of the circulating white blood cells. These guys have a very large round nucleus, so much so that you can sometimes just barely see the cytoplasm around the nucleus. These guys continuously migrate from the bloodstream to the tissues and then back to the bloodstream. There are 3 functional classes of lymphocytes. There are T cells, or you can also call them T lymphocytes. These are part of our cell-mediated immunity, meaning that they can attack foreign cells or they can attack our cells when our cells become invaded by viruses. There are B cells, or B lymphocytes, which are part of our humoral immunity. So these are the cells that actually make the antibodies that I introduced you to earlier in this particular chapter. And then you have natural killer, or NK cells, and these cells provide immune surveillance. One of the important things they can do is that they can help to find precancerous cells before they turn into full cancer. So they act as kind of a surveillance to see if any cell looks abnormal or out of place. And we're going to go into much more detail on these 3 in chapter 22. So we're just introducing them to you at this point. In this section, we're going to look at leukopoiesis and its regulation. So leukopoiesis is another term for the production of white blood cells, just like erythropoiesis was the production of red blood cells. So remember from earlier in this lecture, we have the hemocytoblasts that are in the bone marrow or myeloid tissue. And the hemocytoblasts divide to become myeloid stem cells and lymphoid stem cells. And then we also talked about myeloid stem cells, which can divide to become different types of progenitor cells. And then last time we proceeded and talked about red blood cell development. Now we're going to talk about the white blood cell development and production. So some of those progenitor cells are going to become what we call myeloblasts. The myeloblast is going to give rise to basophils, eosinophils, and neutrophils, which are those 3 that were classified as granulocytes. Some progenitor cells become monoblasts, and the monoblast is going to give rise to the monocyte. Monocytes enter the bloodstream while they are still immature, and then they are technically not fully matured until they leave the bloodstream to become macrophages. Then, if we go back and remember that we had myeloid stem cells and lymphoid stem cells, the lymphoid stem cells become lymphoblasts, and the lymphoblast will eventually become lymphocytes. And lymphocytes get their own name for their own production. Lymphocytopoiesis is the process of lymphocyte production. By now you should notice that the esis part on the end is talking about production, and the name in front of it is telling you what cell is being produced. So the lymphocytes are actually formed in the bone marrow and in the lymphatic tissue. So some have to leave the bone marrow and finish their development in other types of tissues. For example, the thymus gland is where T cells go. That's why they're called T cells, T for thymus. But we're going to get more about that in chapter 22. So leukopoiesis, or white blood cell formation, is regulated by hormones called colony-stimulating factors, or CSFs. These are hormones that regulate the production of white blood cells. There is a multi-CSF, which actually will regulate not only the granulocytes and monocytes, but also platelets and red blood cells. So the regulation is way up at the level of the myeloid stem cell, and the myeloid stem cell becomes granulocytes, monocytes, platelets, and RBCs. The GM-CSF regulates the production of just the granulocytes, which are going to be your neutrophils, eosinophils, and basophils, and the monocytes. And then the M-CSF is only going to regulate the production of monocytes. And so colony-stimulating factors can be released by macrophages, lymphocytes, and even endothelial cells. Endothelial cells are the cells that line blood vessels, and so they release these colony-stimulating factors in the event of an injury or infection, and that is going to stimulate you to make more white blood cells. That is why, like, when you have an infection, your white blood cell count will go up temporarily. So here is our nice little summary here. So again, we started at the top with the hemocytoblast. They became myeloid stem cells and lymphoid stem cells. The myeloid stem cells became 3 different types of progenitor cells. One of those progenitor cells goes on and follows the red blood cell pathway when it's stimulated by erythropoietin, and we talked about that in a previous section. One of the progenitor cells goes into the platelet pathway, which we haven't talked about yet. And the last progenitor cell goes into myeloblast and monoblast. The myeloblast becomes your basophils, eosinophils, and neutrophils. The monoblast becomes your monocyte. And then the lymphoid stem cell becomes your lymphoblast, which becomes your lymphocyte. So notice where the colony-stimulating factors are in these pathways. So multi-CSF is regulating at the level of the myeloid stem cell. That's why it can also affect platelets and red blood cells. Your GM-CSF is only at this level, so it is only affecting basophils, eosinophils, neutrophils, and monocytes. And then you've got the M-CSF, which is just affecting monocytes. And we actually didn't include this one on the list, but the G-CSF would only be affecting the granulocytes. So up here, G and M stand for granulocyte, monocyte. G here would just be granulocyte. M here would just be monocyte. And then we come to our final formed element, which are the platelets. And we'll talk about their function and their production. So platelets are not actually cells, but rather pieces of cells or cell fragments. They are also called thrombocytes because they are involved in blood clotting, and a blood clot is also called a thrombosis. Platelets are continually replaced so that each one only spends about 9 to 12 days circulating through the bloodstream before it is removed and replaced. So the spleen we've talked about before. This is the site of phagocytosis of old platelets. We talked about it previously because it also is where you have a lot of phagocytosis of old red blood cells. So the spleen is an excellent recycler of our formed elements. And the spleen also stores 1/3 of all of our platelets. So people who have had their spleen removed do not have a larger reserve of platelets to pull from in a time of emergency. Another piece of terminology to know is thrombocytopenia. This is when the platelet count is too low, and this would make it harder for a person's blood to clot. So you have a normal platelet count shown here. You don't have to remember the numbers. And you can see the platelets in the bloodstream amongst the red blood cells. And then with thrombocytopenia, you would have a reduction in your platelet count. So platelets function to release chemicals importing to the clotting process, and we're going to look in detail at the clotting process in the next section. Platelets also form a temporary patch in the walls of damaged blood vessels. This is called a platelet plug, and this slows the blood loss while the clotting takes place. So here's a picture of a platelet plug that has formed in this tear in this blood vessel. And platelets can also reduce the size of breaks in the vessel walls. So platelets do contain bits of actin and myosin filaments that can contract, just like we learned last semester when we talked about the physiology of how muscles work, and muscles shorten because of the interactions between actin and myosin. Well, a similar mechanism takes place here, but in this case, when the platelets contract, they pull the broken edges of a blood vessel closer together, and so that's going to reduce the size of the break. So let's look at platelet production, and it has its own name. Thrombocytopoiesis would be the production of platelets. So here is a picture of the pathway. So we're going to start with the hemocytoblast like before, which is going to become a myeloid stem cell, which is going to become a progenitor cell. So some of those progenitor cells are going to become these very large cells called megakaryocytes. So these are huge cells. That's where it gets this name, megakaryocyte. And after they're produced, they're going to spend a little time making structural proteins like that actin and myosin, enzymes, and extramembrane. And then it's going to start shedding off bits of the cytoplasm in small membrane enclosed packets. And these are what platelets actually are. So platelets are little membrane enclosed packets of a bit of the cytoplasm from a megakaryocyte. So it's going to have some actin and some myosin in there. It's going to have some clotting enzymes in there. It's going to be surrounded by membrane. One megakaryocyte can make about 4,000 platelets. And things that affect platelet production: there is a hormone called thrombopoietin, or TBO, which is a hormone produced by the kidneys. That should sound familiar. There is also another molecule called interleukin-6, or IL-6. This is an inflammatory hormone, which is also called a cytokine. It can be secreted by macrophages and endothelial cells lining the blood vessels. And then multi-CSF, we talked about in the previous section. This is the one that is stimulating the myeloid stem cells. So anything a myeloid stem cell becomes is affected by multi-colony stimulating factor. And so this would stimulate the megakaryocyte formation and growth. In this section, we're going to talk about blood clotting, which is also known as hemostasis. So don't confuse that with homeostasis, which is something completely different. This is hemostasis. Whenever you see the prefix heme, you're talking about blood. So hemostasis is composed of 3 phases and then a final clot retraction. And before we start, we're going to be using the term endothelial cell a lot. So I wanted to remind you, you were introduced to these back in chapter 4 when we talked about different types of tissue. And we talked about simple squamous epithelium. And we said that the simple squamous cells that line blood vessels are called endothelium. And so they are also known as endothelial cells. So endothelial cells are the simple squamous cells lining blood vessels. So phase 1 is the vascular phase. So this is going to be the first phase that occurs when a blood vessel is injured. So damage to the blood vessel walls are going to cause smooth muscle contraction. We're going to look at the structure of blood vessels in chapter 21, and you'll see that most of them do have a layer of smooth muscle. So they have some smooth muscle in there that can contract. And this is called a vascular spasm. And the purpose of this is to narrow that blood vessel to slow or prevent further fluid loss. The endothelial cells lining the vessel also contract, and the endothelial cells start releasing chemical factors and hormones. One of these is called endothelin. This is a peptide hormone that plays a key role in this whole process. Your textbook does mention a bunch of other chemicals and factors involved in this process. If I don't talk about them in the PowerPoint, I'm not going to test you over them. So endothelin stimulates smooth muscle contraction further, and it also is going to stimulate cell division for repairs. So it's going to get the endothelial cells ready to divide so they can replace the damaged endothelial cells. Same thing with the smooth muscle cells. You're also going to have fibroblasts in the area that are going to get ready to make repairs, and they're going to need to divide. So basically we're getting ready for repairs at the same time that we're slowing the fluid loss. Endothelial cells also become sticky, so they release these hormones that make them stick to each other, and it makes them stick to any platelets that happen to be coming by. And that leads us into the second phase, which is the platelet phase. So platelets are going to come by and they're going to adhere to those sticky endothelial cells, and this is called platelet adhesion. So the platelets adhere to the damaged vessel. Then more platelets are going to arrive and they're going to stick, and they're not only are they going to stick to the endothelial cells, they're going to stick to each other, and we call this platelet aggregation. And all of these platelets sticking to each other is what's going to result in a platelet plug. And this is a fairly fast process. This can begin within 15 seconds of an injury. The platelets, as they come and get stuck to each other, this causes them to become activated. And when they're activated, they release clotting factors and other chemicals. And once they release these clotting factors and other chemicals, that gets us into our third phase, which we'll talk about in just a second. But first, I also want to mention that this is one example of positive feedback. When we first learned about negative and positive feedback in A&P 1, you learned that most systems in the body work by negative feedback and very few systems work by positive feedback. Well, this is one of them. So a platelet sticks to the damaged vessel wall. That platelet becomes activated. It releases chemicals that are going to attract other platelets, which come and stick. They get activated. They release chemicals, which is going to cause other platelets to come and stick. And then they get activated and they release chemicals. So it's a positive feedback mechanism that's just going to cause more and more platelets to start aggregating and stacking up. So because a positive feedback mechanism can technically get out of control, you know, at some point I could just have too many platelets that are stacking up and adhering to each other. So I have to have some way to put on the brakes. So there is a compound called prostacyclin. This is a type of prostaglandin that is produced by the endothelial cells, and it helps to inhibit the platelet aggregation. So it helps to keep this phase under control so that you don't just get runaway platelet aggregation that lasts forever. So then once the platelets are releasing those chemicals, we move into the coagulation phase. This is when the actual clot is formed. So this involves very complex clotting factor cascades, and your textbook goes into great detail. And for those of y'all that are going to nursing school, I think eventually you're going to have to learn all the steps of the clotting factor cascades of all three pathways. We are not going to go that deep this semester in this course. So all I need for you to know as far as the coagulation phase is that there is an extrinsic pathway. This takes place outside the bloodstream in the vessel wall. There is an intrinsic pathway that takes place inside the bloodstream. And then the two converge together at what is called the common pathway. You can see here it's called a factor X, where they come together. And this results in the production of an enzyme called thrombin. So of all of the different factors listed here, I want you to know thrombin. And what thrombin does, thrombin converts fibrinogen to fibrin. So that's the ultimate goal of all of these pathways, the extrinsic pathway, the intrinsic pathway, the common pathway. The ultimate goal of having this pathway is to get that thrombin, and the thrombin converts fibrinogen to fibrin. So I introduced this to you earlier in this lecture. Fibrinogen is a soluble protein, but when thrombin acts on it, it is converted to fibrin, which is insoluble. So fibrin is actually like a bunch of little threads that are going to appear in the area. And then once those threads appear, they can trap other elements like red blood cells, white blood cells, debris. They can trap anything in those fibers, and that is what is called the blood clot. So after the clot has formed, the platelets will contract, and this will pull the edges closer together. This is called clot retraction. If you have a wound on like your hand or your finger, sometimes, after a few hours or the next day, you can notice the edges of the wound look puckered up, and that's because of the clot retraction. And this also not only helps to reduce fluid loss, but it helps pull the edges closer together to make repair easier, so that the endothelial cells and the fibroblasts and all of the cells that need to work to repair the area don't have as far of an area that they have to stretch over as they're repairing. Eventually, though, you're not going to need the clot, and then so the very final stage would be fibrinolysis. This would be the destruction of the clot after the repairs are complete. And again, your textbook goes into all of the specific enzymes and chemicals that are necessary for this. But if there's a chemical or enzyme in your textbook chapter that I did not mention in the PowerPoint, you don't need to know it. So some terminology related to clotting problems. Hemophilia is a genetic disorder caused by mutations in some of the clotting factors. So people with hemophilia can't make some of the key clotting factors. So they have a hard time producing blood clots, and even small bumps or bruises can turn fatal from uncontrolled internal bleeding. They can also just spontaneously start bleeding, and of course it gets very dangerous if they even have like a small hit to their head that can cause runaway bleeding in the brain, which can be fatal. Hemophilia is usually only in males because it's passed along the X chromosome. And one famous example are a lot of the royals in Europe back in the turn of the century or the turn of last century. A lot of members of the royal family had hemophilia, including the last Tsar of Russia. His son had hemophilia. Thrombophilia is an increased risk of forming clots. So it's like the opposite. Hemophilia, you can't clot. Thrombophilia, you clot too much. And if you have a clot occur when it shouldn't, it can be dangerous. So basically, you don't want the inability to clot because that's dangerous, but you don't want to make too many clots, especially when you don't need them, because that can also be dangerous. So for example, a deep vein thrombosis, or a DVT, is a clot that can form in the venous system, and these usually form in the legs, and we'll talk a little bit more about this in chapter 21. But the danger with a deep vein thrombosis is that it can become dislodged, travel through the bloodstream up to the lungs, and get stuck. And this can cause a pulmonary embolism, which is a blockage of the pulmonary arteries from one of these clots. And these are very serious conditions. They can be fatal 30% of the time. And of course, blood clots can also be the cause behind strokes and heart attacks. So you definitely don't want clots when you shouldn't need them. And things that can affect hemostasis. So I mentioned earlier that hemostasis works by positive feedback, so you can get runaway clotting if you're not careful. You have to have something to put the brakes on. So we do make natural anticoagulants, and two of them I've already mentioned to you. Heparin is an anticoagulant released by basophils and mast cells, and prostacyclin is a prostaglandin released by endothelial cells, and your textbook gives you several other molecules released by endothelial cells, but I'm only going to tell you about those two. Other things that can affect hemostasis: aspirin. Aspirin is an anticoagulant, so it slows down the clotting process. This is why people at risk for heart attacks take low-dose aspirin every day. It's actually to reduce their risk of having a clot, because it can reduce your risk of, or actually reduce your ability to clot. I take a lot of aspirin because I also have migraine headaches, and so I bruise very easily. And again, that is a side effect of having an anticoagulant, is your blood is slow to clot. Vitamin K is required for the formation of several clotting factors, including prothrombin, which is what turns into thrombin, and thrombin was that key enzyme that converts fibrinogen into fibrin. You get about half the vitamin K you need from your diet, and the other half comes from the bacteria in your large intestine. If you have insufficient vitamin K, this can cause a breakdown in the common pathway and eventually deactivate the whole system. You can get vitamin K from like dark leafy greens like spinach. You don't really typically have to worry about vitamin K unless you're not eating a good diet, or if you're taking antibiotics for a long time and it's killing the bacteria in your large intestine. If you notice from the previous picture that I showed you of the coagulation phase, calcium ions are used in multiple steps. So this process absolutely requires calcium ions. So if you have a condition like hypocalcemia, which is too little calcium in the blood, this will adversely affect your ability to form blood clots. So we will wrap up with a quick summary of the formed elements. So here is a table from your textbook. So it lists all of the formed elements. So you have your red blood cells, then you have your white blood cells, your neutrophils, eosinophils, basophils, monocytes, and lymphocytes, and then you have your platelets. So it tells you the abundance, and notice that these numbers here are per microliter, which is a very small volume. It shows you how they would appear in a stained blood smear. Then it tells you their function. So this is the column that we talked a lot about. And then we talked a lot about the remarks over here as well. So you can read these to help you review the material. And then here is a summary of the production of the formed elements. All formed elements start with the hemocytoblast in the red bone marrow. The hemocytoblasts become myeloid stem cells and lymphoid stem cells. The lymphoid stem cells only become lymphoblasts and then lymphocytes. The myeloid stem cells become 3 different types of progenitor cells. One of these, when it's stimulated by erythropoietin, will change to a pro-erythroblast, will then go through several erythroblast stages. At the normoblast stage, it will eject its nucleus. It will become a reticulocyte, it will enter circulation, and within about a day, it will become an erythrocyte or a red blood cell. A second progenitor cell will become a megakaryocyte, which will shed its cytoplasm to make platelets. And a third progenitor cell, when stimulated by a certain colony stimulating factor, will become 2 types of blast cells, myeloblast and monoblast. The myeloblasts go on to become basophils, eosinophils, and neutrophils, and the monoblasts go on to become monocytes. And then I've got one more section here. You have enough A&P knowledge at this point to be able to read your blood test results and know what the items being tested are. And then the test results that they let you have also will show you what your value is compared to what it should be. Of course, if anything doesn't match up, it's up to your physician to interpret that and determine a treatment plan if needed. But at least now when they do give you your blood lab reports, you can at least read them with some knowledge now that you've gotten this far in A&P. So first, a couple of terms. Venipuncture is the collection of whole blood from a superficial vein. This is the most common method. This is how they're going to take your blood when you go to the doctor or go to the hospital. Superficial veins are very easy to locate, especially the ones in the inner part of your elbow where they typically do take your blood. The walls of veins are thinner than arteries, and we'll talk more about that in chapter 21. And the blood pressure is lower in veins than in arteries, and we'll talk about that in chapter 21 as well. So again, the typical venipuncture site is going to be in the crook of the elbow. There is a vein there that is the most common vein used for blood draws. There is an arterial puncture, also called an arterial stick, in which blood is drawn from an artery, but this is usually only done if you need to analyze the blood gases and specifically oxygen levels. So now I'm going to show you what some of the common blood tests results are on your lab reports. This information is for you only. This is not going to be on the exam, but I thought that you might be interested to know how to read those lab reports that you get from your doctor. Some lab reports may appear slightly different because it depends on the units that they put on the lab report. And unfortunately, my doctor doesn't put the units on theirs, so mine is not as detailed as I would have liked, but still the purpose of this is to show you what the different items are. So a complete blood count, or CBC, provides information about your blood cells, aka your formed elements. So here's what a typical report would look like. You have the name of the test on the left, you have the result value in the middle, and then on the right you have the normal range. And like I said, I would have liked it if they'd put the units on here, but the most important thing is comparing your actual value to the normal range anyway. So the WBC is obviously your number of white blood cells, red blood cells, the amount of hemoglobin you have in your blood. The hematocrit, you should know by now, is the percentage of formed elements in your blood. So MCV, you might not be aware of, that stands for mean corpuscular volume, and that is the average volume or size of a single red blood cell. So they're checking to make sure your red blood cells are the correct size. MCH is mean corpuscular hemoglobin, and that is the amount of hemoglobin in a single red blood cell. MCHC is mean corpuscular hemoglobin concentration, and that's going to be the average amount of hemoglobin in a certain amount of red blood cells. So they're really checking to make sure that not only are your red blood cells the correct size, but they have the correct amount of hemoglobin inside of them. RDW is red blood cell distribution width, and this is a measure in variation of size or volumes of red blood cells. So how similar are all of your red blood cells to each other, or do you have some that are standing out, like that are too tiny or too big? Platelets is obviously going to be your number of platelets. MPV is your mean platelet volume, and this is going to be the average volume of a single platelet. Lymphs would be your number of lymphocytes. Monocytes would be your number of monocytes, or these are percentages, sorry, percentage of lymphocytes, percentage of monocytes, percentage of neutrophils, percentage of eosinophils, and percentage of basophils. And now you know what all of these cells are and what they do. And the CBC can also show an absolute count of the different types of white blood cells. So on the previous page, it was showing the percentages, and then they can get an absolute count by taking the percentage and multiplying it by the overall number of white blood cells, and that will just give you an absolute count number for each of the 5 types of white blood cells. And then I thought I'd also show you a little bit of a metabolic panel. Metabolic panels provide information about fluid balance, levels of electrolytes, and how well the kidney and liver are working. So here's a sample metabolic panel. Glucose would be your fasting blood glucose levels, and we learned in chapter 18 that those need to be within a certain range, or it could indicate that you have diabetes. Sodium would be the sodium ion concentration in your blood. Potassium would be the potassium ion concentration in your blood. And so again, in chapter 18, we looked at hormones like aldosterone that can regulate the levels of sodium and potassium. Chloride would be the chloride concentration. Carbon dioxide is how much CO2 you have in your blood. And again, notice that getting an oxygen level would require an arterial stick, and they don't do that when you go into the doctor's office for a routine blood test. They do put that little oxygen monitor on your finger to get a rough reading of your oxygen level that way, but it's not going to show up on your lab reports. Calcium would be your calcium ion concentration, and again in chapter 18 we looked at how that was regulated. So we talked about calcitonin and parathyroid hormone and how they work together to keep calcium within normal ranges. Total protein, you now know this is those plasma proteins. Remember, they make up about 7% of the volume of plasma. So now you know what that means. Now you should also know what albumin is. That is one of those plasma proteins. It is the one that is in the highest percentage of protein, is is going to be albumins, and they function as transport proteins. We also talked about globulin earlier in this lecture, and so they measure that as well. They also do a ratio between your albumin and globulin. And then finally, bilirubin. So we talked about that this lecture as well. The bilirubin comes from your red blood cell turnover. Remember, this is ultimately what those heme groups are broken down into, and then it is released into the bloodstream, and then the liver has to filter it out. So this is a good measure of if your liver is working correctly. If your bilirubin amount is too high, if the value is too high, that can be an indication that something's wrong with the liver. And then I didn't show every test. I left off some of the common kidney function tests and the liver enzyme tests since we haven't covered those organ systems yet. But I wanted to show how much you already know and how much you should be able to even read something like your lab report from your routine blood tests. And that is the end of Chapter 19. Make sure you start studying now.